बच्ची बहुत घट गई एंड झट मार दे मड़ी क्या
So, I now request our distinguished session chair Dr. Sunil Kaul and our organizing chair Dr. Pati to please kindly honor our session lead Professor Radha Kanta. I now hand over the session to our distinguished session chair, Dr. Sunil, whose leadership will steer the further proceedings of the session. Thank you, sir.
So, good afternoon and welcome back. So, this is a technique session, and as I the Dye Lab, which is an international laboratory, and as you see that it says AIST India Dye Lab. This is the only laboratory in Japan which says India. So we are very happy and proud about it. And under this Dye Lab program, we've been running different programs, and one of them is Picnic. Picnic stands for Platform for Innovative Knowledge to International Know-how. So under this program, we come to different universities in India or elsewhere, and then organize a session with different expertise. So this is one of the picnic program that we are having today. So, as you can see, the picnic speakers this time. So, these are Renu, you can identify here. I do not know which year it is. 1979. Okay. 1979. Dr. Gurucharan is here. And Dr. Avinas is here. So, all the three big pillars of this. So, picnic program we've been doing since 2014, and we had about 6 big picnic programs at GNDU so far, and this is going to be the 7th one today. And as you can see from our previous picnic programs, that we had one in 2017, Picnic Series 16. So you can see Dr. Doi and our members here with a big group here, GND group. And Picnic Series 24 was held in 2018. Then we had 2020 and 2022. And last year again we came here for Picnic Series 64, and we are again here at 26 today here. With this, I would like to invite now Dr. Renu Wadhwa, the first speaker. She needs no introduction. She's a globally recognized molecular biologist and very popular alumna of this university. So, Renu, please.
Thank you so much. Thank you so much for the lovely gestures that I'm receiving since morning. I'm really moved. Thank you, Pratap. So, Picnic has been introduced to you already. So my purpose for the next 17 minutes, as given, is to introduce to you a little bit of science that the lab has been doing. So today I'm going to present to you stress and cancer connection. Somewhere we all know they are connected, right? We know a lot of stress will give you disease, it might give you cancer too. But me here standing in front of you is going to present to you a laboratory evidence using cells as a model where we prove that an extreme level of stress leads to cancer. The mild stress, when you boost your body systems, might be good, average stress, you might be able to cope because you have the inbuilt mechanisms to cope with these, but the extreme stress will lead to cancer. So this is the topics my lab has been working for last 36 years on: stress, aging, brain diseases, neurodegeneration, and cancer. As I introduced, I'm sure everybody knows what cancer is, and it can affect any part of the body. The most critical for the students is when cancer affects you in a way that your normal cells forget to stop to divide. You know, they just forget they need to stop. They keep on going, dividing, and that's what cancer is. So that can happen anywhere, any tissue. And keep in mind that I'm going to present stress and cancer. They are huge fields. I'm going to present to you the connection between two with the experimental data that we have published. So we have been working on this field for nearly 3 decades, using cells as a experimental model system. We compare normal cells with the cancer cells and try to see what proteins are overexpressed, what proteins are underexpressed. Then we try to see how they are contributing to the mechanisms of cancer or stress or that matter for aging. 2 proteins were cloned in my hands. One is named as Morlin, which is stress protein, and the other is CAF. That's a topic of my talk today. Just shortly, I want to tell you that both of these proteins, they are associated with the dividing or proliferation phenotype of cells. The Morlin and CAF levels go down, the cells die. They go up, the cells keep on dividing. So let's focus on CAF. So it happens in the lab that we were trying to look for the proteins that bind to a tumor suppressor protein called P53, and we found this new protein. That time it was new, in 2000, I think it was 2000. So this new protein, new cDNA, we try to see what function it does in the cells. And you see here when the CAF is overexpressed in the cells, along with another tumor suppressor, the colony size goes down and the colony number also goes down. That means this new protein, which we called CAF, for the purpose that it is a collaborating member of AF, collaborating partner of AF, like this. So this is tumor suppressor. So this induces growth arrest into the cells. That means the stopping the growth or stopping the proliferation of cells. We try to validate that into the different cellular model systems. For example, we know the cells will stop dividing when they age or when they senesce. Yes, CAF levels go up, and it replicates the upregulation of P53 and P22, the well-established markers of growth arrest or senescence or aging. So we found this new member here, CAF, which is also upregulated. Okay, that's fine. So what happens if you induce the cells to age? You can do that in the culture system. You stress them and you have them stop dividing. If you do so, your CAF levels also go up. This is in response to hydrogen peroxide. Or the other way around, if you overexpress CAF, the cells will stop dividing. So that proved all these experiments, taken about 2 to 3 years of time in one slide, showed that CAF is a tumor suppressor. Very fine. So we thought, if it is a tumor suppressor, if we knock it down, cells should be immortalized. They should live longer. They should proliferate for longer time. So we did that. So we knocked down now CAF in normal cells and in the cancer cells, and none of them lived longer. Rather, they enter apoptosis, proving that this is something which is essential. If it is not there, cells just die. They enter the process of apoptosis there, and so then we actually proved that how does CAF function. This is a triple labeling of the cells. You see the new centromere or the kinetochore as green, you see red as a membrane and blue as a DNA. So this was the normal one. You saw that the 2 cells divided into very nice, beautiful daughter cells. But if the CAF is gone or compromised, let's see, doesn't divide. It just stays there and it will burst into apoptosis. So that was the molecular mechanism we addressed, and you can see up there as well. Control cells will divide into 2 nice daughter cells, and if the CAF is compromised, Cells are there, they are undergoing the process of division but never complete it. They just burst into apoptosis. So that proved that calf is very, very essential for cells to live and proliferate. If it is not there, the cells die. And remember, I showed you the data. If it is overexpressed, it is a growth arrest or the aging. So we are interested to see how does it function. What is the mechanism? So we made in the lab the cells which are now expressing serially upregulated levels of calf here. This is GFB tag. So you can have the cells with showing serially increasing level of calf. Very surprisingly, we found that overexpression was decreasing the growth, but the superexpression was making them malignant. Just try to think what I'm saying. The overexpression was stopping their growth, as I showed you in my back slides. But if it is superexpressed, 10 times higher level, the cells are now not stopping, but they are just malignantly transformed. In other words, it is cancer. So this was very surprising, but a field for us to work on. We now proved yes, superexpression of calf, this is superexpression, change the epithelial morphology to mesothelia. Another criteria for cancer, increase the migration capacity of cells, criteria for cancer, and increase their invasion capacity. Another capacity or another criteria for the cancer cells, again proving that the superexpression was linked with the cancer, carcinogenesis, or malignant transformation. Let's see here. Now, if we compromise calf to the level that the cells are not apoptosing or cells are not dying, we wanted to see if that would decrease their migration. Yes, it did. That would decrease their invasion. Yes, it did. So calf compromising can actually a way to decrease the migration or invasion. In other words, it was contributing to these phenotypes. Now we also checked the in vivo system. Here there's a calf super expression. You see the higher or the increased growth here, and knockdown is now decreased growth. Similar thing here, you see the metastasis is not happening if the calf is knocked down, and here the tumors are not even growing. So calf is associated with the carcinogenic phenotype. So in the human databases, cancer patient databases, again, calf is upregulated, and that was seen in a large number of tumors. Here is one example. So over the years, took about 10 years to publish all this. We described the mechanism, how the overexpression is leading to growth arrest, and how the superexpression of calf is leading to malignant transformation or carcinogenesis. So that was nearly 10 years of research in less than 10 slides. So if you are interested to see the more, please look at the papers. But in any way, so I'm trying to relate how stress would lead to calf superexpression, and that in other words means cancer or carcinogenesis or malignant transformation. So we wanted to know what kind of stress upregulates calf. So we used 16 models of stress in the lab listed here. These are the chemical models of stress. You can actually stress your cells in the culture dish and see how response is there. This is a summary of the full paper we have published. When calf is decreased, the cells enter cell death or apoptosis straight away. When there is no change, the cells just were happy being there. And when there is overexpression, they enter growth arrest or premature aging. And when there is superexpression, the cells enter malignant transformation. Just like what I showed you in my last slides, same thing reflected here. And I just show you example data. Here, no change is a control. And then when this increase, overexpression, there's a growth arrest, or senescence or aging, premature aging. Then when there is a super expression, there's large number of colonies here, is the malignant transformation in the other ways. So that means calf is bridging the biology between stress and carcinogenesis. We want to know again basic biology, and we're still working on it, how this overstress or super expression of calf is linking the stress phenotypes to the malignant transformation. The message is clear, but we know beyond doubts that when there is a super expression of calf driven by super level of stress, it is going to give you cancer, right? So another parallel stream that we would like to do is to intervene the level of calf or intervene the level of stress in the other ways, so that you don't get to the level of cancer. You are stressed, but the bridge between cancer and stress you want to break. So this is a parallel project where we see the compounds which are active for protecting your cells against oxidative stress, metal stress, and hypoxia stress. And I think you will agree with me that these three stresses together makes all sort of neurodegeneration or the other related diseases: oxidative, metal, and hypoxia. So this is a triple cell screening method that we developed. We expose the cells into these three series of stresses and screen for the compounds which are now protected. And amazingly, we found the top hits were from ashwagandha. This was another, parallel project going in the lab, but it's done in the different hands and in the parallel. We found that ashwagandha compounds, vidofludimus, vidanone, methoxyvidofludimus, and others, they are protecting the cells from this three-way combined stress screening assay that we developed. So, in the lab, of course, in the last 20 years, we already published, yes, ashwagandha is got a good anti-cancer activities. Now I'm saying it is also protecting against stress. It does make sense. If it would protect you against stress, of course, it will prevent cancer or protect you from cancer as well. So the anti-cancer activity of ashwagandha has been extensively published, and we have published that vidanone and vidofludimus are the active compounds in ashwagandha which can protect you or which can be potential anti-cancer drugs. So now we are focusing on anti-stress activity, and I'm going to show you some data. The cells which are put into premature aging by a compound called methyl acetic acid are protected here if vidanone is there. Now the cells, normal cells, not cancer cells here, the normal cells exposed to oxidative stress and UV are protected here. Again, the cells which are cultured with low doses of vidanone are living longer as compared to the control cohorts. And here we are again seeing that the reactive oxygen species created here by stress are now going down. Either you have them by metal or by have hypoxia stress. So anti-stress activity of ashwagandha compounds became clear. Now let me go back and tell you again the link between now these to the CAF levels. When you have the oxidatively stressed compounds or the metal stress cells, the CAF levels go up, and when you protect them with the ashwagandha compounds listed here, CAF levels come down. So we are able to manage super expression of CAF, which was malignant transformation, to the lower levels, which may be growth arrest, which is still better than the cancer. So similarly, here we have the marker for DNA damage, goes up with the stress, comes down, and with that the CAF levels parallel these phenotype here. So this means the ashwagandha that we all very much aware of, we are very proud of this herb, being from Indian Ayurvedic system. The low doses would protect you against stress, then the high doses would treat you from the cancer phenotype. And there are other activities also in this herb we have published extensively, and all this is detailed in the published papers. You are very welcome to look at those papers and look into the mechanisms. Also, there are books on ashwagandha. There are 2 books where we have detailed the mechanisms. Besides ashwagandha, for the intervention of stress, cancer, looking at marker as a CAF level, we've been looking at honeybee propolis, Halictus augustifolia, which is Chinese ginseng. Bitter cucumber is Karela or Goya in Japanese. Wakame is a seaweed herb. Cinnamon, soybeans, leopard lily, and alpha-lipoic acid and Amelia ribes are our favorite herbs put into the research on stress and its link to cancer using CAF as an assay system. With this, I stop here. I think I'm earlier than 17 minutes, and these are large number of students who have actually done and contributed to the work. My thank you goes to them. Thank you very much for listening.
Thank you, Renu. So we'll take questions at the end or during tea time. We move on to the second speaker.
So we have the privilege of inviting Dr. Javeed Ahmed He is the director of CSS,